Dr. Sage here. In this set of 2 video lectures, we're going to talk about the evolution of populations. By the end of this first video, you should be able to realize how population genetics is used to study evolution, explain the Hardy-Weinberg principle and its importance, and calculate frequencies using the Hardy-Weinberg equation. So first, some basic definitions. A population is a localized group of individuals capable of interbreeding and producing fertile offspring. A gene pool consists of all the alleles for all the loci in a population. In this first video, we're going to mostly focus on the Hardy-Weinberg equation. Okay, the Hardy-Weinberg principle describes a population that is not evolving. So if a population is in Hardy-Weinberg equilibrium, it means that population is not evolving for that particular trait you're looking at, that particular gene you're looking at. If a population does not meet the criteria of the Hardy-Weinberg principle, it can be concluded that the population is evolving for that trait. So, Hardy-Weinberg principle is used to determine if a population is evolving. The Hardy-Weinberg principle states that frequencies of alleles and genotypes in a population remain constant from generation to generation. In other words, if it's not evolving for a particular trait or a particular gene, then the amount of the certain allele you have, let's say big A versus little a, should remain the same from one generation to the next generation. In a given population where gametes contribute to the next generation randomly, allele frequencies will not change. And Mendelian inheritance preserves genetic variation in a population. Now, there are 5 conditions that must be met for populations to not be evolving. First, there must be no new mutations. Okay, changes in DNA that create basically a new allele. Second, you must have random mating. Okay, the population must not be selecting for a specific trait over another trait, like selecting specifically brown fur over white fur. That must not be happening. It must be random. Third, there must be no natural selection. So there must be no selective pressures for that particular trait. Fourth, it must be an extremely large population size. Why? Because when you have a very small population, it's very easy to, just by chance, randomly drift out of that particular genetic frequency. Fifth, there must be no gene flow. Now, these last 2, extremely large population size and no gene flow, I'm going to explain those in much greater detail in the second video in this set. So we'll come back to those topics. Using Hardy-Weinberg, the frequency of an allele in a population can be calculated. For diploid organisms, remember that means 2 of each chromosome, therefore 2 of each type of gene, the total number of alleles at a particular locus or particular gene is the total number of individuals times 2. Why? Because every individual has 2 copies of every gene. The total number of dominant alleles at a locus is 2 alleles for each homozygous dominant individual plus 1 allele for each heterozygous individual. The same logic would then apply to recessive alleles. So by convention, if there are 2 alleles at a locus, we're going to use P and Q to represent their frequencies. And we're going to use P to represent the frequency of the dominant allele, and we're going to use Q to represent the frequency of the recessive allele. So, since you have a dominant and a recessive, the frequency of all alleles in a population must add up to 1. So therefore, P plus Q must equal 1. So in this example, let's say we're talking about the pea plants that Mendel had. Remember, in pea plants, you can have a yellow pea or a green pea, and yellow is dominant over green. So capital Y would be the dominant yellow pea color, lowercase y would be the recessive green pea color. Okay, so the frequency of the dominant allele, capital Y, is going to be represented by P. Frequency of the recessive allele, lower y, is going to be represented by Q. Now, since there are only 2 alleles at that particular locus, P plus Q must equal 1, or 100%. Okay, so as an example, let's say we have a population of peas where yellow is completely the dominant over green for pea color. In this population of peas, we have 245 yellow homozygous dominant peas. Remember, that would be capital Y, capital Y. 210 yellow heterozygous peas, so capital Y, lowercase y, and 45 green homozygous recessive peas, so little y, little y. So if we want to calculate the number of copies of each allele. Capital Y would be 245 times 2, because you have 245 homozygous dominant P's, and each of them has 2 copies of the Y. That's why it's times 2. Plus 210. Why? Because we have 210 heterozygous P's, and each of them has 1 copy of the Y. So 700 total copies of that capital Y dominant allele. Now we calculate the recessive allele, little y. It'd be 45 times 2, because we have 45 homozygous recessive P's that have 2 copies of the little y, plus 210 heterozygous P's that have 1 copy of the little y. So that adds up to a total of 300 copies of the lowercase y. Okay, so that's how many numbers of each of the different alleles we have, big Y versus little y. Now to calculate the frequency, which is what P and Q represent, P is the frequency of the dominant, in this case the capital Y. Well, as I just told you, there were 700 total copies of that capital Y, divided by 1,000 total copies of that particular gene, both the dominant and the recessive. So if you have 700 divided by 1,000, it's 0.7 or 70%. Now, Q represents the frequency of the recessive allele, in this case little y. There are 300 total copies of the little y in that population, divided by a total of 1,000 copies of that particular allele, whether it be dominant or recessive. So 300 divided by 1,000 would be 0.3 or 30%. And as I mentioned, P plus Q must equal 1. 0.7 plus 0.3 equals 1, or 100% total. All right, so that's how you start to use the Hardy-Weinberg equation. Okay, the math is not that difficult. It just takes a little while to get used to what P and Q represent, how you calculate P and Q, and remember P plus Q must equal 1. However, we're not going to stop there. That was the frequency of the 2 different alleles. Well, let's say we want to know the frequency in a population of the homozygous dominant, or of the heterozygous, or of the homozygous recessive. Okay, well, since P is the frequency of the dominant allele, Q is the frequency of the recessive allele, then the second equation that goes with Hardy-Weinberg that you must know is P squared plus 2PQ plus Q squared equals 1. Okay, now what does that mean? P squared and Q squared represent the frequencies of the homozygous genotypes, and 2PQ represents the frequency of the heterozygous genotype. Okay, let me explain that again. What P squared is the frequency of the homozygous dominant individuals, the ones that have 2 copies of the capital Y. What Q squared is the frequency of the homozygous recessive alleles, the ones that have 2 little y's. Okay? So if you have homozygous dominant and you have homozygous recessive, the other category would be heterozygous, 1 big Y and 1 little y. That is 2 times P times Q. And again, since there's 3 different possibilities, homozygous dominant, homozygous recessive, and heterozygous, those 3 must add up to 100%, or must add up to 1. So P squared plus 2PQ plus Q squared equals 1. All right, so for example, in those P plants, remember we calculated P, the frequency of the dominant allele, as 0.7. We calculated Q, the frequency of the recessive allele, as 0.3. Okay, well, given that, let's say what we now want to know is that what is the frequency in that population of homozygous dominants. Remember, homozygous dominant is P squared, so you just simply take P, which we already calculated was 0.7, and you square it, which equals 0.49, which what that means is 49% of the population is homozygous dominant. Similarly, let's say we want to know homozygous recessive. That's Q squared, so we simply take Q and square it. So that's 0.09, or 9% of the population is homozygous recessive. The one that's left is the heterozygotes, big Y, little y. That is 2 times P times Q, so 2 times P times Q equals 0.42, or 42% of the population is heterozygote. And then remember, P squared plus 2PQ plus Q squared must equal 1. 0.49 plus 0.42 plus 0.09 adds up to 1, or 100%. Okay, so that's how you use a Hardy-Weinberg equation. And you can verify it using a Punnett square. Yeah, so if we have 245 in the population that is homozygous dominant, plus 210 that is heterozygote, that'd be 245 times 2 plus 210 equals 700 total copies of that dominant allele. Then we have 210 that are heterozygous, so 210 copies of the recessive allele, plus 45 that are homozygous recessive, so 45 times 2, 90 copies of that recessive allele. That adds up to 300. 700 of the capital Y's divided by 1,000 total, that's 700 plus 300, equals 0.7, which is the P, or 70% is the dominant allele. Then you have 300 lowercase y's divided by 1,000 total equals 0.3, which is the Q, and that's the recessive allele. So this is just figure form of what I started to show you. Now, let's say we want to know the frequency of homozygous dominant, that's 2 big Y's. Well, that would be P squared. So P, 0.7 times P, 0.7 is P squared, 0.49. You can verify that in the Punnett square. Okay, let's say we have Q is 0.3 and Q is 0.3, so the chance of being homozygous recessive, it would be Q times Q, or Q squared. That would be 0.09. Now, for the chance of being a heterozygote, you have a capital Y and a little y. So the chance of a capital Y is P, 0.7. Chance of a little y is Q, 0.3. 0.7 times 0.3 is 0.21. But remember, there's two different ways you can get that. You can get that up here, or you can get it down here. 0.7 times 0.3, P times Q is 0.21. So that's why a heterozygote is 2 times P times Q, because there's two different ways you can get that. So 2 times 0.7 times 0.3 is 0.42. 0.49 plus 0.42 plus 0.09 equals 1. Okay, so we just verified what we calculated using the Punnett squares. You can see why we get the numbers we get. So, this is a biology class, but you're gonna have to do a little math in this biology class. Honestly, this is about the extent of the math we're gonna do in here. You need to learn how to use a Hardy-Weinberg equation. Make sure you memorize what P represents, frequency of the dominant allele. What Q represents, frequency of the recessive allele. What P squared represents, frequency of the homozygous dominant. Q squared, frequency of the homozygous recessive. 2PQ, frequency of the heterozygote. P plus Q equals 1. P squared plus 2PQ plus Q squared equals 1. As long as you have one piece of information, if you have P, you can calculate everything else. If you have Q, you can calculate everything else. If you have P squared, you can calculate everything else. If you take P squared, you just take the square root of it, now you have P. Since you have P, you can calculate Q, because P plus Q must equal 1. So as long as you have one piece of information, you can calculate everything else. Best thing you can do is practice some of these Hardy-Weinberg equations to make sure you understand it. Because in math, you need to practice math to understand how to use math, okay? So you need to be doing that for this course, because there's going to be Hardy-Weinberg equations on your exam. You're going to have to be able to figure things out. Okay, so when a population is in Hardy-Weinberg equilibrium, the allele frequency is stable from generation to generation, and the distribution of alleles can be determined by the Hardy-Weinberg equation. If the allelic frequency measured in the field differs from the predicted value, then scientists can make inferences about what evolutionary forces are at play. So if a population is not in Hardy-Weinberg equilibrium, then evolution is happening. Now, how can it happen? That's what we're going to talk about in the next video lecture. We're going to talk about population genetics and adaptive evolution. So, until next time, this has been Dr. Sage.